Maybe. All right, so I've hit the record button. So we're going to do a brief short lecture on cell signaling. And so if you're following along with the stuff that I put in Canvas for you, this is on the cell signaling basic notes that you have in there. So you can grab that if you wish. I'm going to kind of jump around because for me to sit here and lecture you about this stuff, you don't grab enough of the information. You've got to work through it on your own. You've got to be active in that. So the Quizlet's available for you. The Bookit is available for you. I would suggest you do those. And remember, it's short amount of time, lots of different times. Okay. So like when you guys were gone to cheerleading thing, there may have been times when you were just sitting around doing nothing. You were bored, mad at everybody. Pull out that phone, spend 5-10 minutes doing one of the Quizlets, and then just stop. And then if you get bored a little later, do the same thing. Just make sure that you're reviewing things that you studied before you start the new material, okay? Standard thing. I learned a little bit, take a break. When I come back to do it again, I review, and then I add another piece to it. And you just keep that process up, and that process would be, that process is what active learning really is all about. Multiple times, short amount of times with review. All right, so we're going to go through this here real quick. First of all, what do you think of when we say cell signaling? What do you think of?
Signal communicating cells.
Yeah, there's a signal. Something is communicating. Something is happening between the cells, right? If you've ever been or seen a movie where they have an airplane and you got the pilot, but then there's a guy on the ground with flags and they're doing, different things with those flags. You guys probably don't know much about racing, but in racing, it's a big deal. There's always a flagman, and the way they hold the flag and what color the flag tells the drivers. Like if there's 2 laps left, they hold up 2 white ones and they tend to move them back and forth. A white flag means it's the last lap. It's a signal that happens. It's a way of communicating, okay? There are 2 major systems in animals that do signaling. One system is the nervous system, and one is the endocrine system. Which one do you think works faster? In other words, yeah, the nervous system, absolutely. You guys got it. And the endocrine system is longer. If I were to scare you, or as a coach, I used to try to hype up the team before we actually played the game. What I'm trying to do is get the adrenal gland to dump adrenaline or epinephrine, depending upon what side of the pond you're on, into their system. That usually takes 8 to 10 hours to get back to levels prior to when it started to rise. Now, granted, it's a nice spike and it does drop off relatively slowly at the end. But that's why after you get really scared and the issue is gone, you're still kind of, and it just takes a while for you to relax and calm down. And that's just because of that hormone. What system does the adrenaline or the epinephrine, depending upon where you come from, what systems do you think that molecule affects? Body system? Cardiovascular system. What else? The lungs, the respiratory system. Those are your 2 biggest ones. We're going to dilate those blood vessels. We're going to increase respiration because we need to make a decision on whether we're going to stand and fight or whether we're going to run away. Does that kind of make sense? Okay. We need to break down how that works. And that's the basis for what we're doing today. We're going to break that down on how that works. So there's 3 broad pieces to this. The first piece is called reception. So whether or not it's a long distance thing, in other words, your brain sees danger and then a signal is sent to the adrenal glands via the nervous system, and the adrenal gland is down on your kidney, dumps out adrenaline into your bloodstream, okay? That's a long distance one, or whether they're right next to each other, you still have to have reception, okay? And so you can see the definition there. It's the target cell's detection of a signal. So it's the detection of a signal. Okay? That's all it is. And for us, especially between cell to cell when it's long distance, we have a thing called a ligand. And the ligand is just any molecule that does that. So in my example with adrenaline, my ligand would be the molecule adrenaline. It would attach to the outside of cells, and then there would be some kind of effect that it would have. Okay, so more often than not, they're going to test you where the ligand comes from outside the cell, not inside the cell. So please make sure that you go back and look the difference between intercellular communication and intracellular communication. It's part of those Quizlet vocabulary things. You guys had it on your list, and definitely that was a good thing to do. The second stage of this, if you will, is called transduction. And this one is pretty straightforward, but it's basically everything that happens after reception until you get a response. Okay, so again, you can look on the notes that are in there. It says the conversion of the signal to a form that can cause a response. So conversion of the signal to form until forming a response. Until forming a response. All right, so the adrenaline attaches to a point on the outside of the cell, or any of the ligands do. That causes change inside the cell. That means you might have, and I'm just going to kind of make a little bit of a membrane here. So let's say you have some transmembrane protein. So if you don't remember your membrane proteins from last semester, please go back and look at them. I can help you out with that, okay? You want to make sure that you've got that. So what may happen is some molecule, some ligand attaches, that causes changes, which then causes reactions to occur. And usually those reactions are a series of reactions. So everything from after the ligand attaches all the way down till you get your response, that's transduction. Yes, sir.
Are these proteins specifically for signal transduction?
Yes. And a lot of times there's only one or two molecules that will actually fit. Remember, these proteins, many of them are going to be enzymes. And so they're going to have an active site, and they're going to be either lock and key or induce fit to a specific molecule. Okay? Now, what's kind of cool about some of this stuff is we can create chemistry, pharmaceuticals, to literally lock things up. Like when you go to the dentist and you have a tooth that's got a cavity in it and they hit you with that lidocaine, they're literally shutting down all the pathways of pain that are going in, and they're blocking, because there's a competitive inhibitor that blocks the protein, so you don't get any transduction, so you don't get any response from those cells. That sends pain signal back to the rest of your brain to be processed, and it's, oh, that's pain. And then as those molecules come out, those ligands come out of these things, you start feeling the pain again. You no longer have this transduction block. But it's really, really important for your project. We've got reception. So here you want to look at enzyme substrates. There's going to be some of that. And you want to look at membrane proteins. So you want to look at those two things. Go back and review those two things, because that knowledge comes forward. All right, and then the last, we have reception, we have transduction. I've kind of hinted at it. The last one is going to be response. Last is going to be response. Now, this can be a lot of things. And AP Bio usually picks about 4 is what they pick. One of them is called apoptosis. It's called cell death. The cell receives a signal and it terminates the cell. What happens inside the cell with transduction causes the cell to be terminated. Okay? One of them that I think is really kind of cool is that the signal comes in, the cascade comes down, it goes down to the actual DNA. And remember, your DNA, if you don't remember from biology, but your DNA is all twisted up and locked down. And so when you need a protein made, there are chemicals that go down and they unlock that particular section, open it up. We do transcription off of it. We make the protein off of it. There's literally signal pathways that cause your DNA to be switched on or switched off. And there's a lot of medicines that do that. If you ever want to be a pharmacologist, sure, work in a pharmacy at a hospital or a private practice or whatever, or a CVS, you'll probably go crazy. But if you start doing the research and stuff, that's what these guys are looking for. They know the biology and they're trying to get certain genes maybe to switch on or certain genes that are on and shouldn't be and switch them off. That's your response. What happens because of that signal? Okay? So you got to know those 3 things: reception, transduction, and response. Okay? Now, here's what you need to make sure you do. Go through and look at the basic piece, and also look at, I've given another file for you, another page to look at. It's the 3 primary stages. And then after that, for each stage, I have a specific one that talks about the different kinds. For your project, you're going to get a signaling pathway on your project. So far, so good. And so those files will be very helpful because they target specifics. Do not go and memorize every one of those 3 things. There's like 10 in each. That's like 30 different very specific pathways. They give you a lot of information on the exam, but you have to decode it. Does that kind of make sense? You've got to decode what's going on. And your first piece is to start with the reception, transduction, and response. And what you guys are going to do in your groups is look at very specific ones. All righty? Okay, that's it.